real world scenes. And we find that even in natural videos, the same GNN architecture can generalize to match human judgments much better than the non-graph model. So it seems like, contrary to what's been found previously, as long as you add the right inductive biases, which I think in the case of social interactions is the fact that they are inherently relational, you can match social judgments with purely visual models. And I think this really suggests that relational information may underlie our abilities to recognize social interactions. So just to summarize, we found that social interactions are selectively processed in the human brain, in the human STS, separately from all sorts of other visual and social information like faces, voices, theory of mind. And that we think relational and visual information in particular may underlie human social interaction representations. And these visual computations are what allow us to so quickly and effortlessly recognize social interactions. So you can go to our lab website or follow me on Twitter to get more updates about this work. And I am happy to take any questions.
Thank you, Leila, for the nice talk. So is there any question from the in-person audience? I guess we have one like a minute for a question.
Hi, Leila. Do you think that the social interaction, I think, is also an area that you correct me if I'm wrong, that's also important for biological motion? Is there a way to separate out those two things?
Yeah. Yes. So good question. It seems like in both our controlled study and the movie study, they are very close by, but separable from each other. So in the point light stimuli, for example, these two, both the interacting ones and the pair of independent agents have roughly equal amounts of biological motion. And while it's very difficult to measure biological motion in a natural video, most of the motion energy in a model is actually due to people moving around. So we think that that was a pretty good control for that as well. But you're right that biological motion is also processed very nearby in the PSTS and also an important social function. And so I think it is interesting to think about how these two types of information interact with each other.
Yeah, that's interesting because they're probably very close together. And yeah.
Yeah.
Cool. Thanks.
Thanks.
Nice. So yeah, if you have more questions for Professor Isik, you'll have more chance during the panel discussion. So thank you for the talk. On to the next talk by Professor Baylor from UC Santa Barbara. Mike, yeah, you can go ahead.
Awesome. Thank you to the organizers for having me. Very excited to be here. And thank you to all of you for sticking around. I'd like to switch gears a little bit and tell you about an exciting application at the intersection of visual neuroscience and computer vision. This is one example where, as a computer vision researcher, you definitely need to know about the neuroscience behind it in order to make a meaningful contribution. And the application I'm talking about is the potential to restore vision to people who are blind with a neuroprosthetic device. Now, as you might be aware, there are millions of people worldwide that live with irreversible vision loss. There are many different causes that cause vision loss. Two of the most common players are retinitis pigmentosa, where you slowly lose your vision from the periphery inwards, leading to tunnel vision, or age-related macular degeneration, where you lose vision in the fovea and the macula first, and then it spreads outwards. Although there are no cures for these diseases, researchers have been trying to come up with ways to treat them. Some of the current approaches that are being studied are gene therapy. And because these diseases are caused by a faulty gene in the retina, the idea is maybe we can replace the faulty gene with a functioning one and thus reverse or at least slow down the disease. A second approach is to use optogenetics to make neurons responsive to light that have never responded to light before. And that allows you to shine a laser on them and activate them precisely. And the third approach, which is what I want to talk to you about today, is the idea of implanting a microelectrode array, either directly into the eye or into visual cortex, to stimulate the surviving neurons with electrical current. Now, this is no longer science fiction. The most famous device, perhaps, is the Argus 2, developed by a company called Second Sight. This device is sometimes also referred to as the bionic eye. It comes with a pair of glasses and a small embedded camera that records the scene. And then frame by frame, the visual scene has to be translated into a series of electrical pulses that are sent wirelessly here with this coil to a receiving coil implanted in the eye that then stimulates the microelectrode array. We can zoom in on this a little bit more to see here the different layers of the retina, and then the implant sitting right on top of the ganglion cells. This particular implant is equipped with a 6x10 electrode grid, each 200 microns in diameter, roughly. So that makes each electrode orders of magnitude larger than an individual cell, something to keep in mind. This device has been implanted in 300 to 400 people worldwide. It is currently discontinued, but there are over 20 new prostheses currently in development. So the field is expected to only grow over time. And even though there are all these different devices in development, some of which will be implanted in the eye and others directly in visual cortex, they all share some common components. And very quickly, they all have an external camera, usually, which is used to acquire visual input. This signal is then sent to a vision processing unit, so usually a mobile device that can do some image processing and stimulus optimization. And then that is sent to the microelectrode array, typically wirelessly through transmitting and receiving coil. Then what happens if we stimulate this device? We will electrically activate neurons in the brain, which is how they usually communicate. So if we do this smartly, the brain might not be able to tell the difference between an artificially stimulated neuron and a naturally stimulated neuron. At least that is the idea. So stimulation of electrodes will produce some neural activity that is then interpreted by the brain as a visual percept. Now, many years of research have focused on this pipeline here. So how do you go from an applied stimulus to the predicted response, either neural activity response or a perceptual response? And what people have done is kind of boiled down the visual processing of the brain into what we call a forward model, basically a black box model that maps stimuli to responses. Perhaps the simplest forward model is just a linear model. We call it the scoreboard model, because it assumes, like maybe you've seen scoreboards in sports stadiums, it assumes that every electrode is kind of an independent light source. And so if you stimulate a grid of electrodes, that should lead to the percept of a grid of luminous dots. If you only have 10 by 10 electrodes, then the produced image might not look very interesting. But if we increase the number of electrodes, eventually we should be able to produce useful vision. Now, this has been used in combination with some computer vision techniques, because obviously what you need to do now is you need to encode a complex visual scene such as this with a low resolution display. And so many different ideas have been explored, such as perhaps we can end-to-end optimize the encoding. If we have a target image, we can learn how to encode that with these small phosphenes, as we call them. Perhaps you want to do it like this, or you want to just predict the edges. Others have thought about, no, maybe what is really important is the ground plane. If you're a blind person, you have to find your way around town. It's important to know where you can walk and where you cannot. So maybe you want to segment the ground plane first and then visualize that. Or maybe you want to combine segmentation with structural edges, which can give you an idea of the room layout, and then combine that into a phosphenic representation. Or maybe it is more important to just highlight nearby obstacles, because that will eventually allow you to react to your immediate surroundings. So many different computer vision techniques have been explored, but all of these suffer from one crucial limitation, which is that this way of producing these phosphenes is not biologically realistic, meaning that these phosphenes cannot explain some of the distortions that have been observed clinically. We know this because we have tried it out. We have worked with real patients and asked them to draw on a touchscreen what they see when we turn on a single electrode. This particular person here, when we turn on one of their electrodes, they would draw a line. And that seemed very odd at first, so we repeated it. And we see that over many trials, the person would always draw the same line very consistently. So what we can do is we can align all these images by their center of mass and average them to get a good idea of what this particular electrode, here called D3, would produce. Here's another example. These are all vertical lines aligned by the center of mass and averaged. You get a pretty good idea that this particular electrode always produces a vertical streak. Repeating this for all possible electrodes in the array, we find that all of them produce different streaks, but they're repeatable across trials, right? Because this is averaged. You see that over time, the percept is repeatable. It's just that going from one electrode to another, the orientation of the line changes drastically and seemingly randomly. This is just one patient. So repeating this experiment for different patients, we find a whole repertoire of different phosphene shapes. They include lines and triangles, wedges, all sorts of things, but most of them I would not characterize as a pixel or a blob. And so where do these distortions come from is a big open question in the field. We could either spend 10 years doing psychophysics to have a data set that's large enough for a deep net to fit, or we could dig in a little deeper into the neuroscience and see if we can find underlying causes of these distortions, as we call them. So to give you a really quick crash course of the retinal physiology, you might already be aware that when light enters the eye here, it gets refracted and projected onto the back of the eye, which contains the retina. And the retina already is a maybe 3 or 4 layer network that processes the incoming visual signal. We have our photoreceptors, the light sensitive cells, that then send their signal to bipolar cells and finally to ganglion cells, which are the output layer of the retina. And they send their signal through these axons that go to the optic nerve to the back of the head where the visual cortex sits. Now, if you have one of these blinding diseases, such as retinitis pigmentosa, what happens is you lose your photoreceptors. They start to degenerate. And so the idea here is to replace the lost functionality with a chip. If you put it right there, it's called subretinal, but there are other locations where you could put your device. The one I just showed you, the Argus 2, that's called an epiretinal device. And you can see it sits here on top of the ganglion cells. But not just the ganglion cell bodies, it also sits on top of the ganglion cell axons. And that turns out to be the key for these phosphenes. Here is now a bird's eye view of the back of the eye. And every one of these lines is a fiber bundle, an axon that has been extracted from 55 cadaver human eyes and aligned by the optic disc and the fovea. And what you see is that these fibers form a very stereotypical pattern. So now we can, I guess, ignore the math. We can explain this with a spiral with a very small number of parameters. And we can start to ask what would happen if we had an electrode right there and we stimulated that neural tissue. What would happen is we wouldn't just stimulate the ganglion cell bodies that live directly below the electrode. We would also stimulate the ganglion cell bodies that send their axons through this point in space to the optic disc. And all of these happen to fall on a line, because if you're a neuron that lives over here, you will send your axon along this pathway. And so this one, same for this one. So by activating them all at the same time, we're telling the brain that we saw something and it was right here, but it was also right next to it and right next to it and right next to it. So essentially what we're doing is we're producing a line, a streak. There's another structure landmark in the retina that's important. That's the horizontal raphe. That's a region where there are no axons. The reason for that is if you are a neuron that lives directly above it, you will send your axons here, up here, and over to the optic disc. And if you live right below it, you will send it downwards. Okay, so skipping many years of research, we were able to come up with a computational model that can predict what a phosphene will look like depending on where it sits on this axon map. A schematic is shown right here. If you now stimulate a certain electrode, it is assumed that the current will spread not just radially, it will also spread along these axon bundles. And we can parameterize the spread with two parameters, rho and lambda. So if you make rho larger, if you make rho larger, you should get a blobbier percept. And if you make lambda larger, you get more of a streak. But that's not all. The shape of phosphenes also depends on the stimulus parameters. For example, if you increase the amplitude, phosphenes tend to become more blobby. And if you increase the frequency, the shape doesn't change, but the brightness does. And then finally, if you change the pulse duration, you can get rid of some of the streakiness and turn the phosphene more blobby again. So all of these are nonlinearities that are not taken into account by the traditional scoreboard model that a lot of people tend to use. And therefore, predictions with your scoreboard model might be highly misleading. To help people understand the quality of these phosphines, we have developed software called Pulse to Percept, which is a Python package you can all download and try out. And it can predict for all these common retinal implants that are out there, what the vision generated by them would look like. I wanted to show you a couple examples here for fun. So here we're simulating what an Argus 2 might produce. Remember 6x10 electrodes? If you were in the subway and you see people walking by, without this axonal activation, you basically would just get a downscaled version, grayscale version of your input with a 6x10 grid. But as soon as you add the axonal activation, things start to kind of smear out and get elongated, which you can appreciate most here. Again, maybe 6x10, that's just not good enough. So what happens if we increase the number? For reference here, again, 6x10. What if we now had a 12x20? And you can start to make out more details of your immediate surroundings. But it's also evident that you get kind of this fisheye effect, right? This local distortion and bending of the individual pixels, if you will. So there's an opportunity here to optimize the stimulation, not just go pixel by pixel, but incorporate knowledge about the distortions to produce better stimuli. And so going back to the schematic I showed in the beginning, here's the setup where you go from stimulus to response. What we really want is not this forward model that tells us for a given stimulus, what is the response. We want to know the opposite. We want the arrows to flow in the opposite way and tell us, how do I achieve a target response? And so one way to solve this or to think of this is in an autoencoder structure where your forward model is fixed because it's informed by neuroanatomy, behavioral results, all sorts of things. And what you want to learn is an encoder that inverts this forward model such that some reconstruction error is minimized. To make it clear in the application of a visual prosthesis, you would start with a target percept, it could be an image or a movie, whatever. And you would feed it through an encoder that you now want to train such that the stimulus it predicts, if you feed that through your forward model that is fixed, it should produce a percept that looks just like your target percept. We call this a hybrid neural autoencoder because the forward model is typically a symbolic or a numerical model informed by the neuroscience. And the encoder is just a deep net that is trained to invert this fixed model. Often it's impossible to invert this fixed model because it's either too computationally expensive or it is not differentiable. So some people instead use a surrogate approach where they train a deep net to approximate the forward model and then use that in an end-to-end fashion. So we wanted to see how both of these approaches work, one where you have the fixed decoder in the loop, and one where you use a surrogate model, and compare it also to the naive approach. As a reminder, naive approach just means whatever image you have, you basically paint it on the retina. And then depending on your rho and lambda, the output will look better or worse. For example, if rho and lambda are really small, then yes, you basically have this pixelated model that a lot of people assume we have. But we also know some patients, they have much larger lambda, so much streakier percepts. And if you just apply the number 3 there, then you get something like this, and it no longer looks like a 3. And the same is true if you increase your rho. So different patients have different values of rho and lambda, meaning that this naive approach only works in the most ideal cases. However, the hybrid neural autoencoder seems to perform quite well in a range of these different rho and lambda values. Here tested on the MNIST data set, because everyone loves MNIST now. So here we have some target images and some results for small values of rho and lambda. You can see the HNA is performing really well, producing a crisp image that fits the target very well. But the naive is not doing so bad either, as pointed out before, because rho and lambda are small. Interestingly, the surrogate model, where we trained a deep net to approximate the forward model, is already performing badly. And we will see that gets even worse as we increase, for example, lambda here. So now in the naive model, you should see long streaks. You also see it in the surrogate. But if trained end-to-end, the HNA, the hybrid model, can learn to get rid of these streaks that you would get from a naive encoder. It learns to compensate for them. And so the same is true as you increase rho in the bottom row, Greek rho in the bottom row, and lambda as well. So you can see in the naive encoding here, you just get a blob of light basically, whereas the hybrid encoder can still perform pretty well. To show you that this also works somewhat on more complicated data set, we also tried on MS COCO. And here to help the network a little bit, we segmented the main object in the scene as shown here. So this is the target image. And then you can see the prediction of the hybrid model and the bottom row, the naive prediction. And so while the hybrid models is not perfect, there's a lot of detail is lost, we did some additional studies to show that this is mainly a limitation of the number of electrodes. Here we used 15 by 15 electrodes. And if you increase the number with the same model, you actually get a much crisper, much better result. So this seems to be a very promising strategy in general. But I personally believe that this doesn't go far enough. To tell you a little bit more about how these devices are typically used is that each company comes up with their own stimulus encoding, how to go from video to stimulation, electrical stimulation. Often this is proprietary because they might use some fancy computer vision image processing. But then there's this one encoding that is being validated on different external tasks. So basically there's a hope that this provided encoding, this provided vision will be good enough to support tasks such as visual acuity, object recognition, or navigation. And instead what happens is that people say this artificial vision is very different from natural vision. And often it's not good enough for user expectations because they have to spend weeks and months of training to be able to use this new visual language. So instead, what we're proposing is that the optimal encoding might be highly context dependent, right? So if you are interested in visual orientation, like finding identifying objects in relation to you, then you might rely on one encoding, and that might look very different from if you're doing a mobility task and you're just trying to get from A to B. And it might be even more different if you're just interacting with people. There are different visual augmentations that could help here, right? Some might include object segmentation, others might include highlighting nearby obstacles or faces. But the bottom line is that there should be different modes that focus on functionality. So rather than making any claim about restoring vision, we should provide a tool set where the user can choose the appropriate task, the appropriate encoding for whichever task they're engaging in. And so this would move away from clinical tests and focus more on the real world tasks and find the visual augmentation strategies that optimize performance on specific everyday tasks. And so we call this the smart bionic eye, because it would provide AI-powered visual augmentations to support specific tasks. Again, if you are outdoors and you're navigating, maybe it's just important to know where nearby obstacles are, maybe cars and people, but you also need to kind of have an understanding of where you are in relation to your surroundings. So perhaps there is a computer vision-based pre-processing strategy that would allow a prosthesis user to perform well in this particular task. How do you find the best strategy? That's really hard. So what we propose or are already doing is we're using a virtual reality prototype. Because we have this forward model, we can have a system that shows you the simulated prosthetic vision in a VR headset. And so then we can have sighted users act as virtual patients and perform the tasks we're interested in while we try out different augmentation strategies. In the end, this focuses a lot more on the user and the end user experience. And so it's moving more towards an iterative design process with the end user in the loop, such that you can really find the thing that maybe doesn't look the best, but it provides the most functionality. You're all aware that there are a myriad of strategies that could be useful here, since deep learning can operate on single RGB images to segment objects or to even estimate relative depth, for example. There's methods to find the structural edges of the room layout, and there's things that haven't been explored in this application at all. For example, visual question answering, where you could ask, for example, where did I place my keys again? And while you look around with your bionic eye, it would detect the keys and then outline them for you. So once these computer vision systems get more sophisticated, there are a lot of exciting avenues to pursue. I just want to make the point that it is less about dreaming up these new augmentation strategies, because there's a lot of them. And it's more about really identifying the visual cues that best support behavioral performance. A quick example most of you might be familiar with is optic flow. So we know that in texture-rich environments, we rely on optic flow to navigate, but we're also able to switch to different strategies in less cluttered environments. And for example, navigate more by landmarks. And these abilities change with vision loss. So how can we know which cues, which way to augment the scene is going to be most supportive for these particular end users on a particular task? Then finally, our implementations have to be efficient, because they have to go on the visual processing unit, and they have to be robust, right? Because if you miss an oncoming car, it could be a life or death decision. So it really requires designing systems with predictable failure modes, not just working at the edge, but really being able to allowing us to understand how they make the decisions. So that goes into explainability, but I also understand when and how they fail exactly. And so to wrap it up, I believe that computer vision has the potential of drastically improving the artificial vision generated by a bionic eye. But however, to provide meaningful solutions, we need to understand the neuroscience. And that might involve using these realistic phosphene simulations that are psychophysically validated, such that our predictions can be more or easily translated to the real world use case. And there's an engineering challenge with designing efficient and yet robust systems that have predictable failure modes so that we can use it in the real world. With that, I say thank you, and I'm happy to answer your questions.
Thank you, Michael, for the really interesting talk. Is there any question from the audience? Or from Zoom attendees? I have a question. So you use of this prosthetic device, will it require training of the user? Like, what does an EDS mean, or what does a blob mean? Is it an EDS or is it a deft map? I mean, they may mean different things to different people.
Yes, it will definitely require training. A lot of people describe their NeuroVision as kind of like fireworks, fireworks show. You see these flashes of light that appear, and they seem very unnatural first. And second, require you, therefore, to associate certain forms and shapes with meaning. So people have to be trained usually, both in terms of their strategies, like how to sample the visual scene, how to move your head to get the important information. And then also in terms of interpretation, what do these shapes mean and how can I make use of them?
I see. I think Jeff has a question.
Yeah, the transforms you were looking at were very specific to the placement of the prosthetic, right? Because you were placing it on the bundle. So what would be the optimal place to, with being not too invasive, to place this prosthetic? And how would that change what kind of algorithms you'd have to use?
Yeah, that's a great question. I don't think I have the backup slide for this, but we looked at this. In the retina, you don't have cortical magnification for one, so it's a little easier to place the array a little more parafoveally. But with simulations, we looked at what would be the best orientation and location to place the array, and it turned out to be really close to the raphe and basically flat, because that kind of minimizes your streaks. But the answer would, of course, be very different if you now go in V1, where you have this strong cortical magnification. And so currently, you would have to train a different encoder for every location, for every patient, basically. We're trying to overcome that limitation, but yeah, it's hard.
Thanks.
Mm-hmm.
Yeah, very nice. Yeah, really interesting.
Thank you.
Any other question? If not, then next up is, oh, sorry, we have a break now.
Okay.
So
where do the recordings go? Do they go to us, or do they go?
I think they go to CVPR because they generated the links.
Okay.
Yes.
Good. Yeah, because some people were asking, I think some of the people from Europe and India were asking if they could, of course they're going to bed now, so could they see some of the later talks?
Right. Yeah. Yeah, I think, yeah, so we didn't create the Zoom link from our account, so yeah. There is no coffee shop in the, I mean, there are coffee shops, but all are closed already. Some reason.
Conference doesn't have a big coffee station?
They do have a lot of coffee stations.
Oh,
but
that's what's closed. Yeah.
yeah. All right, so it's for welcome all in person and attending via Zoom through to the last session. We have Bruno Olsson, Professor Olsson from UC Berkeley, and he will talk about robust and efficient probabilistic inference in sparse coding networks. So Bruno, you can take it from here.
Okay, thanks a lot. And you can hear me okay?
Yes.
Great. Well, thanks, Hirak and others for organizing and putting this together, and also for having me. I got a lot of the talks this morning, and I apologize for not being able to participate in the discussion. A lot of interesting points came up. I was tuning in, but this being Father's Day, I was driving my father to church and but tuning in. And so anyways, maybe we can revisit some of those things later at the end of this discussion later too. But yeah, so I'd like to tell you about some of our recent work on sparse coding, which is a model of how visual information is encoded in the primary visual cortex. And specifically these properties of robustness and how to do probabilistic inference efficiently. And this is something I think we've made some recent inroads on. And it's mainly the work of my students, Dylan Payton, who worked on the work I'll be telling you about on robustness to adversarial perturbations, and Michael Fang, who's a student who developed this model for doing a probabilistic sampling in the model. So since, hold on, okay. One of the advantages of going later in the program is that you can sort of, in the context of other things that preceded you. So there was a lot of talk in the morning about feedback and recurrent networks in the brain and so forth. And so I wanted to begin by just kind of giving you a feel for why feedback is probably so important in the cortex. And I think this is a property that's just not really been captured yet in any artificial neural network out there yet. And so this is one example of that. It's what's called a puzzle picture due to Dallenbach from the 1950s. And there's a figure in here. There's a figure in this picture. If you haven't seen it before, most people don't see it initially. There's a figure in here, and you can't see it because it's kind of, sort of taken in a somewhat ambiguous way. And so one way of interrogating people what they see is to ask them to draw what they see. And in this particular example, so for one person who was asked to draw what they see, this is what they drew in the lower right. And so it's basically just kind of a veridical representation of the black and white contours in the scene. Okay, so now I'll give you some hints about what's here. It's an animal, and specifically it's a cow. And so maybe for a lot of you, that's already enough to get it. And I'll just point with my mouse here. Hopefully you can see my mouse, but just, so this is the head of the cow in this region here. And there's an eye here, there's another eye here, and the snout down here. So for most people, that's usually enough for it to kick in. And once it does, it's a very strong three-dimensional percept of a cow, and lots of detail emerges that wasn't sort of obvious before what was going on. Okay, so now for this subject, if you ask them to draw what they see, as you can imagine, the answer is going to be quite a bit different. So now this is what they draw. The image is exactly the same. So the only thing that changed is something inside your head. And I think what this importantly demonstrates is that vision and all of perception really is a generative process. Okay, so we're constantly with our brains, this is what evolution designed us to do. We're constantly building models of the world, and we're explaining the incoming sensory data in terms of these models that we're creating in our heads. And in fact, the data coming in is what supports those models, and our ability to explain the data in terms of the model is what supports our belief in these models of what's happening in the world. Okay, so this is really sort of somewhat of a different way of thinking about the vision problem, rather than just casting it in terms of nonlinear regression, training inputs to outputs and doing end-to-end learning and all this stuff. That trying to think of it as a process of model building and inference. Okay, so that's the context of what I'm going to be the work I'm going to be telling you here. And so this is one, this is kind of one way we can schematize the problem. And by the way, I just want to go back and illustrate, mention one thing that I think this also illustrates is the process of explaining away, right? And so what that means is that, for example, some of these details, before you saw the 3D figure in here, you probably just saw it as a loose collection of black and white contours, right? And you didn't really know what the difference between this and this is, right? But now that you know it's here, you would probably attribute this boundary here to being a difference in reflectance, and this boundary to being due to three-dimensional, the shape and the way that the shading is being generated by the light as it's cast upon this three-dimensional object, okay? And so when you say this is due to three-dimensional shading, then you're also saying it's not, or you don't need to explain it anymore in terms of reflectance. And vice versa here, if you explain that in terms of reflectance, then there's no need to explain it in terms of three-dimensional shading. And this is also a huge part of the kind of inferential process is going on in our brain. any given time, there's multiple explanations for what could be happening, and you're trying to sort of rule in and rule out these different possibilities, okay? And that's a process what's called explaining away in probabilistic models, okay? So we're gonna be saying more about that in a minute. Okay, so this is one way of schematizing what's happening is we have the world on the left side, electromagnetic waves that are being focused by the lens onto the back of the eye, creating this image in the back of the retina, which we just heard about in the previous talk, and a whole array of photoreceptors sampling this image. And so the job of the brain then is to try to interpret all these pixel values coming in essentially from the retina, and to try to build some kind of internal model of what it thinks is going on in the world. How do we do that? How do we approach this mathematically? Okay, so the answer for many of us is using the framework of Bayesian inference. Okay, this is not my idea. This is kind of a model idea that's been, sort of propagated in the field now for many decades, actually. And it's since Helmholtz, actually, who described as inference, not Bayesian inference, but the Bayesian inference sort of idea is a little more recent. Okay, but so the idea is we can sort of think that what's going on in the brain basically has a model for how images are generated from three-dimensional causes in the world, from lighting, from reflectance, from how light reflects off of objects and so forth, that we have sort of, somehow we have implicitly of a model, some kind of a model in our brain of how these things are happening and how this light might be cast upon the retina. And at the same time, we have a prior that tells us the relative probability of these different things being out there in the world. So for example, cows are more probable perhaps than other kinds of things that could explain that grainy image. Okay, so the job of inference then is to go backwards and to say, okay, given the image data, what's going on in the world? That's our hypothesis H about what's going on in the world. This is the posterior distribution, the posterior probability that we're trying to develop inside our heads. This is mathematically how you would frame the problem in terms of Bayesian inference. And what's interesting about this inferential process in terms of these graphical models is that even when you formulate the model as a generative model going from right to left, so the model is generating the data, okay, the inferential process inevitably involves this bidirectional flow of information where you have information propagating within layers of representation and between levels of representation. This is just something that unavoidably emerges from the process of inference in these Bayesian models, in the causal generative models. Okay, and so of course, the hypothesis is that maybe these bidirectional flows of activity are what recurrent connections are doing in the brain, horizontal recurrent connections within levels of represented areas and bidirectional connections between levels of representation in the brain. Okay, so that's the overall kind of thesis I'm going to push here. And so how do we sort of drill down on that and make it more specific in terms of the visual cortex? Well, this is an insight that Horace Barlow had many years ago, and from a combination of just looking at the activity in cortex, in which you see activity is fairly silent, only a small fraction of neurons seem to be firing at any given instance, and also just looking at the dimensionality of the representation. So this is an important anatomical fact of the input layer of cortex. So layer 4 is the input layer. You could sort of think of that as like the first layer of your neural network here. That's being indicated by this bar here. And you can just see from this diagram that the number of neurons in the input layer in cortex receiving information from the LGN, which is in turn relaying information from the retina, is vastly greater. So you have many more neurons here in the input layer of cortex than you do, wires coming in. Or these are, you could sort of, again, like these are pixels coming in or wires coming in from the LGN. This is the grain of that representation. Okay. So it's not doing dimensionality reduction by any means, right? That's what the retina is very good at doing. It has, in the retina, by contrast, we have a bottleneck. We're trying to take the information from many cones and funnel it into many fewer retinal ganglion cells. Here's the exact opposite. We're taking relatively few retinal ganglion cell fibers and we're vastly expanding that into a higher dimensional space. Okay, so Barlow reasoned that maybe what we're doing then is taking the input data and carving it up into a feature space, and we're making it very sparse. So these neurons here are very choosy, and only a small handful are going to be firing depending on whether the patterns coming in from the input at any given moment in time match what that neuron is selected for. Okay, so that's the idea that Barlow proposed many years ago, and together with David Field in the 90s, we developed this model, the sparse coding model, which is basically a linear generative model of this process, what sort of hypothesis for how V1 might be processing images. So the idea is that, so what maybe V1 is trying to do is form a very simple generative model. It doesn't have sort of models of cows and faces and 3D shapes and stuff. It's just modeling images in terms of a simple dictionary of shape features, two-dimensional shape features, the so-called Gabor functions, right? So that's what it's trying to do, is fit this model to the incoming data at a given point in time. Then how, if you just sort of work out how inference happens that model, so we can impose sparsity through a prior over these coefficients A sub I, that makes them sparse. This is just the linear generative model of the images. And so when we work all that out and compute a posterior distribution over these coefficients, that leads you to this following minimization problem down here. And when you flesh that as a neural circuit, it leads you to this bidirectional flow of activity among these units. So in other words, it's saying it's not enough to simply, for each of these units, compute the inner product of its feature with the image and then send it to a threshold. That's not enough. You must necessarily talk to it. There has to be this communication between units to form the right representation of the image in terms of this model. So that's illustrated here in terms of this neural circuit. This is something we call LCA, or lateral, the locally competitive algorithm that I developed with Chris Rozelle a while back. And so this is a recurrent neural circuit. It's recurrent inhibition. So each of these units is computing the inner product between its dictionary element, this phi sub I, and the image. And then that's fed into a threshold. And then it's also getting inhibited by the outputs of all these other units. And it feeds that basically the result of that process, the inner product here plus the inhibition from its neighbors to that threshold, and that's resulting activity, that coefficient. And so basically what you're doing, these are the dynamics over here. It's exactly the same as a hop field net, by the way. And it's very simple dynamics, linear leaky integrator being driven by the feed forward projection and getting inhibition from its neighbors, and then going through a thresholding function G. This little g is the thresholding function. So these dynamics solve this minimization problem, okay, for doing Bayesian inference on the image. Again, in terms of a very simple dictionary of features. Okay, so that's the proposal, is you have this very large population of neurons. This is showing a learned population of features, a dictionary for a 16 by 16 pixel image patch. So each neuron is a different patch here, what it's selective to. The pattern that it's trying to find in the image, or it's trying to explain the image in terms of these patterns. And now if you give it a 16 by 16 image patch over here as input, and just compute linear projections. So in other words, if you just compute the inner product between the image and each of those dictionary elements I just showed you, this is what you get. So each pixel here is just showing the resulting inner product for each of those patches we just looked at. Okay, so you can see that's not particularly sparse, actually. So there's a lot of sort of highly distributed activity, and the result of this minimization problem, trying to solve that problem to compute the optimal coefficients, just the ones that are needed, results in this very much pruned set of activities, which then allows you to reconstruct the image. Okay, and so if we go back now and look at that dictionary, which elements did it use to reconstruct this image? You can see it has sort of something vertical here, something horizontal here, something horizontal here. These are the features that it used. So it zeroed out everything else. Everything else here just said, look, I don't need that. And what told it that it doesn't need these things is that it could explain the image in terms of these other features. So this was able to explain much of the pixel variance, and so it didn't need the other ones. And that's the purpose of these lateral interactions. Okay, so this is the process of explaining away. The result is that it gives you a much more interpretable representation, rather than just delivering you the inner product, which is actually very vague and ambiguous. Now it's making a much more meaningful statement. It's saying, look, this feature here is contained in the image. This feature here is contained in the image, and these other things are not. Okay, this is what I think the world is composed of. Okay, so that's basically, so it gives you a more meaningful representation. So it's actually a very sort of important thing that we think is going to be an important ingredient, a kind of important primitive, possibly, to incorporate into deep ConvNet models. Okay, so what's this good for? So I just sort of gave you some kind of hand-wavy verbal explanation, sort of arguments there. But so Dylan was able to show that what this selectivity actually does is that it helps protect against adversarial perturbation. So as we know, this is a big problem that plagues deep ConvNets, is that can be easily fooled by small perturbations in the image. and to miscategorize things. And this is something the field has been working a lot on to understand and to figure out how to make it go away. And so we would argue this is happening because these networks are designed in a very ad hoc way, okay, by just computing linear projection. So let's just look at that, right? If you just think of these arrows here as weight vectors. So this arrow here corresponds to the weight vector of one neuron, and this arrow here corresponds to the weight vector of another neuron in the space. So this is just showing you the input space, the space of possible input stimuli. Let's say for a 16 by 16 image patch. So we're in a 256 dimensional space. Okay, so this is the weight vector of one neuron. That's the weight vector of another neuron. Okay, so now if we want to look at the set of stimuli that activate this neuron with this weight vector here, then that would correspond to these colored lines. And so what these colored lines are showing you the isoresponse contours. That is the set of stimuli that are going to give the exact same response from that neuron. Okay, so if you go to any point along this line that gives the same linear projection onto that weight vector, that's going to give you the same response from that neuron. Okay, so as you can see, it's not very selective, right? A linear neuron is not a very selective mechanism. It's going to respond to a lot of things that are not even aligned to the direction of that weight vector. So when we look at a receptive field of a neuron, we see this Gabor function looks oriented. We sort of think, oh, wow, that's how the neuron gets orientation selectivity. It turns out that's not how it does it, right? That's not good enough. And I'll show an example of that in a second, okay? And so if you wanted to fool this kind of system, and by the way, just thresholding is not going to help you here. If you just take linear projection and threshold, like through a ReLU or something, that's not going to solve this problem, okay? So if you wanted to fool with this system, you could very easily do it. Let's say if I give a stimulus here, and I want to make this neuron respond more, but then I just push out in this direction. So I'm pushing out into a part of the space that has nothing to do with this neuron selectivity, what this neuron appears to be selected for, and that's going to increase the response to that neuron. Okay, by contrast, when you do this process of explaining away through the sparse coding model, when you actually solve this inference problem, when you cast vision perception as an inference problem, even in the context of V1, I'm thinking about these neurons with these dictionaries, this is what it does. It basically curves these isoresponse contours. Why is it doing this? Well, if again, let's just consider these two neurons, right? By the time the stimulus moves over here, so that it's aligned with this neuron's weight vector, well, then this neuron doesn't need to respond anymore. It's basically been explained away. Okay, so that's going to turn this neuron off. So anything over here is going to get inhibited by this neuron here on this neuron's response. Okay, so what that's going to end up doing for this neuron here with this weight vector is just going to make it much more selective than these kind of isoresponse contours. It's going to basically curve these isoresponse contours to make it more selective to things that are aligned with this weight vector. And so here's what would happen in sort of an example of sort of an adversarial attack. Let's say you start with this stimulus, which is a 1 here, the digit 1, and this is what they did here is a simple simulation where you use sparse coding on the first layer and a simple classifier in the second layer to do MNIST recognition. And so if you give it a digit 1 here, for example, at this point in the space, and now you try to fool it into thinking it's a 3, well, it turns out really the only way you can do that is you have to sort of make that digit look more like a 3, right? It's hard to find an irrelevant part of the space to push out into just following these isoresponse contours. So these curved isoresponse contours end up moving you along directions that are more consistent with the thing you're trying to fool it, and so it's harder to fool. Okay, so in order to fool to think as a 3, you kind of have to make it look more like a 3. So this is kind of a neat kind of result they got, and it was published in Journal of Vision in 2020, which I can refer you to for more details. And here's just maybe another simple example to give some intuition by when I say a linear mechanism, something that just simply computes inner product is a very weak mechanism. Here's an example of that, right? So these two stimuli, these two bars here versus these two dots are going to give the exact same response from this Gabor function. The reason why is because here they're relatively low contrast. You have 4 pixels in a row, which are in the excitatory flank, and 4 pixels in a row in the inhibitory flank, versus just 1 pixel here and 1 pixel here that are brighter, right? But so here's a stimulus that's completely, it doesn't really have anything to do with anything we would call this oriented, right? But it's going to give the exact same response from this neuron, okay? And so again, so if you're just computing projection, it's going to be easy to be fooled by that. But in the context of doing inference and explaining away, then these representations become more meaningful. And so we would argue this kind of recurrent lateral inhibition is it's an important primitive of neural computation. It's there in V1. We know, I'm not saying it's doing my model, but we know that right there in layer 4 of V1, there's a huge inhibitory interneuron network. And so this is potentially one thing I would offer that this is my hypothesis of what that network is doing, at least, right? And so this is something we find all over the brain. And maybe this is sort of a useful primitive we could start putting in our networks and to make them more robust. Okay, and there's more to say about that, about recurrent computation, echoing some of the comments of Tim Kreitzman this morning, but maybe I'll save that till later. But okay, so there's another question here I want to address, which is how to do all this efficiently. And so this is another thing I think we have to wrap our heads around if we're going to design systems that even remotely approach the ability of biological systems to do vision, is how do they do this with such low energy at such small form factors? Okay, so I think maybe the thinking that's going on in a lot of people's heads is that, well, I don't need to worry about that now. I'm just going to use my GPU. It's going to consume lots of power. I'll just get my simulation working. And then when I finally get everything working, then I can worry about making it like low energy and small form factor. And well, probably doesn't work like that, right? You probably have to sort of think about those at the same time. And so this is what biology had to do in designing this system. So this is just, I'm just contrasting two things together. The jumping spider on the right, this is 1 millimeter. It has very low energy consumption. And it does remarkable feats of vision. It can do 3D vision. It can navigate in the world. It can hunt prey with his eyes. It moves his eyes back and forth. It does active vision. I mean, it's truly remarkable. If we could simulate the jumping spider on a supercomputer, I think we would all be very happy. We'd be thrilled, right? But we can't. We don't know how to do this. This really still lies beyond our ability to do this, okay? Here's the supercomputer on the left, right? At Berkeley, just up the hill, we have the Lawrence Berkeley Lab, a 5 megawatt supercomputer, okay? And I would challenge anybody to simulate a jumping spider on this computer. I don't think it's going to work. And so, this just kind of, to me, is a huge explanatory gap in our understanding of biology, but it also points the way to opportunity. That as engineers, we should be thinking maybe differently about how to design these systems, that if you need lots of GPUs that are power hungry, maybe you're on the wrong path. We should be thinking about designs and algorithms that can work at this kind of form factor and this kind of energy consumption. And if you don't see a roadmap from your algorithm to that, then probably you're barking up the wrong tree. And so some hints for that, how to design these systems is in terms of these biological principles. There's this beautiful book that came out recently from Peter Sterling and Simon Laughlin. I'd like to point everybody to. This is really sort of like an engineer's handbook for how to design things at low energy and form factor in terms of what the brain is doing. So they're really unraveling and revealing a lot of these principles that the brain is using very specifically. This looks like a very general list, but what they do in this book is they go into very specific examples and retinal circuits in the cerebellum, you name it, all over the brain, and sort of revealing how neural circuits are trading off SNR, signal to noise ratio, with volume, with speed, with power, and so forth requirements in the brain. So it's really, I think, an eye-opening read. And so going back to this thing, so I'm sort of proposing that the brain is doing probabilistic inference. Well, how are we going to do that efficiently? And again, I think one of the important things to remember here is that we actually want to represent a posterior distribution. So in what I just showed you with the sparse coding model, it's actually not representing the whole distribution. It's just finding one point of that distribution. It's going to the maximum. It's finding the most probable explanation. But that's rarely what we really want to do in these probabilistic models. Usually what we want to be able to do is sample or somehow represent these multimodal distributions, okay, multiple hypotheses and their different probabilities simultaneously. And so over the past, looks like, maybe almost 2 decades, there's been this kind of, body of work emerging, starting with this work by Patrick Hoyer and Apo Huvarinen, where we're proposing this idea that maybe this neural variability, this what appears to be noise in the brain, these apparent stochasticity responses in the brain, maybe that's part of a strategy for sampling from probability distributions. Okay, so see, these are a lot of the papers so this is not my novel idea, but we've just sort of advanced this idea more recently in terms of the model of sparse coding. And so I'm running out of time. So I think I'm going to probably just zip ahead and give you the, well, this is not ahead, but I'm just going to maybe end with this point here. This is really the key idea, is that if you represent a probability distribution as a Boltzmann distribution, for example, if you have some like these posterior I'm talking about over the coefficient values, if you can represent that as e to some energy function, then what you want to do, what we're doing right now in sparse coding is we're finding the minimum energy state. Okay, we're just going to this point here. But what we really want to do is sample here. And what's kind of remarkable, so there's this thing called Langevin dynamics, and what it says is that if you want to sample from this distribution, then what you should do is not just gradient descent. So the way we're doing it now is not unlike this. It's very close to doing gradient descent, that LCA, okay? It's not exactly gradient descent, but it's very close to this, okay? So we could design analog hardware, not, other people have actually, we could design efficient analog hardware that computes this very efficiently. People have implemented on Luigi as well. That's not analog, but people have implemented this on Luigi, okay? So we could design hardware that does this very well. But this is what's really remarkable. It says, well, look, if you want to actually sample from that distribution, then just do what you've already do what you're already doing, and just add noise. Okay, so this is just amazing, right? It's kind of mind blowing. So when I sort of learned about this stuff, and so it's saying, look, just build circuits that do this. If you already have a circuit that does this, then just simply inject noise into it, Gaussian IID noise. Okay. And you will be sampling from this distribution. This is actually what you want to do. You don't actually want to go to the bottom and stay here. You actually want to sample from that distribution. Okay. And so this recent paper we have from our group, and that I just gave the reference earlier. So it's the work by Michael Fang. It's going to be coming out in neurocomputation shortly. I'm sorry I don't have time to go put too much detail in it here, obviously. But what this allows us to do now for the first time is learn parameters of the prior in the sparse coding model that we weren't able to do before, okay? So like that parameter lambda that goes in front of the cost function, usually that's a parameter we specify in advance, and we just hold it there. Now we can learn that parameter, we can learn the size of the dictionary. So usually the size of the dictionary is something we have to specify a priori, and we just go with that. So here's what's being shown is you can learn the size of the dictionary. So if you give it a large dictionary and it doesn't want to use all those elements, then they just go to 0. Okay, so this is what happens if you can actually sample from the posterior, then it allows you to fit all these other aspects of the model that normally we've had to hold fix because all we're doing is computing the posterior maximum. Okay, and the big goal that I would have, I think looking forward now, is trying to think about how to build these hierarchical Bayesian models. So sparse coding is a one-layer Bayesian model, latent variable model, but what do we want to have multiple layers of representation? This is Tai-Sing Lee and David Mumford's idea that basically we can think of the visual cortex as a hierarchical model, and they propose that it's doing particle filtering to represent these posteriors at each level. Okay, but potentially now I think, in my view, we have kind of a roadmap for how to move forward with this model to learn the parameters of such hierarchical models. This is some recent work from Matei Lenio's group, a recent paper that came out in Nature Neuroscience, also proposing this idea, not for sparse coding, but for a different kind of model, which is showing that it actually does a good job accounting for a lot of the variability you see in actual cortical responses. Okay, so I think the points I want to leave you with are that these recurrent inhibitory circuits, maybe they're doing sparse coding, okay, and I think there's a lot of evidence accumulating for that. And then one of the advantages this could confer is selectivity, which is going to help protect against adversarial robustness. So maybe this is something we want to adopt in our artificial neural network designs. And finally, that this neural sampling hypothesis, I think, it seems to be becoming a viable hypothesis for how you could represent posteriors, how you could adapt parameters of these models and make them more complex and deep. Anyways, that's what I'll leave you with, and happy to take questions now or later in the discussion.
Hi, okay, I think I got a question.
Yeah, there are a lot of claps. Sorry, maybe you didn't hear it. Thank you very much for really enlightening talk. So if there is any question for Professor Olsson, please, this is the time.
Yeah, I think I got a question. So have you thought about using this sparse coding or like lateral inhibitory approach for maybe like sub-policy networks and reinforcement learning?
What was the first name? Sub-policy network?
Yeah, like maybe like a sub-policy network.
Oh boy, I'm not familiar with enough with that to say, but it sounds like an interesting idea. I'm sorry, I don't. Yeah, I hadn't thought about that.
Okay. Okay. This is really interesting work, though. Cool. I guess I'm gonna go get that book.
What's your thinking about how that might be useful?
Yeah, I'm thinking about how that might be useful for, I guess applying this idea to like creating a generative model for pieces of an image to kind of extending that to like pieces of like motion or things like that.
Absolutely. I mean, I think you could. Everything I presented here was for static images. We're very much thinking about dynamic image sequences, learning motion this way, and those kinds of things as well. Yeah.
I have a quick question. Thank you for the talk. I'm curious, so if you're doing posterior inference with line-driven dynamics on sparse codes, are your samples still sparse like you could have with hard thresholding functions? So if they are no longer sparse because you're adding these Gaussian noise, are you still able to learn these successful Gabor filters?
So the neat thing, what comes out from this dynamics, this is all in the paper. I'm sorry, where are we? Okay, so we can actually now use L0 priors. So this is something that was harder to do before. So usually in sparse coding, people use an L1 cost function. And what you really want to express sparsity is L0, something that just counts the number of non-zero coefficients. But that's impossible to optimize over, right? And so people use L1 as a proxy. And it's not but L1 is not a very sparse prior. So now we can actually use these like priors that are so-called spike and slab priors and do inference in them. And what that gives you is the following dynamics down here. This question is decided. So basically it's just like a leaky integrator with noise. Okay, so just that alone, you're right. Your thinking is exactly right. If you inject noise, that's going to kind of take whatever's sparse and make it denser, right? But importantly, you end up thresholding that. So this is not just sort of some ad hoc thing that we put in. This just emerges as, you know, if you just think about, look at the Langevin dynamics in this setting, you need to threshold it, okay? So you can derive this as the optimal Langevin dynamic strategy. So these variables S now end up being very sparse, and they're sampling from the posterior of using this L0 prior. But yes, you're injecting Gaussian noise here, which is very non-sparse. But what's making it sparse is the thresholding. Yeah.
Thank you.
I have a question. Oh, sorry.
Sorry.
Go ahead.
I have a question. You mentioned extending the sparse coding model into a hierarchical one by stacking these layers. My understanding is that the dictionary being over complete means that your output now is of a larger dimension than before. How do you manage this
Good question.
explosion in size?
Good question. Yes, that's the question. I mean, because if you just sort of make it over complete on over complete on over complete, you know, then your brain is going to explode. So you can't just keep on doing that. So we have some other work called the sparse manifold transform, which I think gives one possible answer to that. And other people have been talking about this too, which is that some kind of combination of expansion and contraction. So you do the sparse expansion, but then you need to somehow contract again before expanding again. You don't just want to take the linear sparse code. I mean, the first layer sparse code and then feed that to V2, for example. So I think that's an important part of it. Also, an important ingredient in there is going to be modeling transformations. So this is again something people have been using either manifold models for. We've been approaching that using Lie groups, and that's another way to sort of condense the representation. So to learn the variability. So this is as opposed to simply doing passive pooling. sort of more model an active process where you're modeling these transformations, and that's another way of condensing it. But you're right. I mean, that's not what your question goes to the heart of the problem, which is why, you know, nobody's really built a stacked sparse coding network that really kind of illustrates the advantages of hierarchy. Actually, there's some very nice work out of Laurent Perrinet's lab, Laurent Perrinet and Victor Boutin. They had a nice paper that came out, and I think both PLOS and neural computation pair papers, looking at what they call predictive sparse coding. So they built a hierarchical model, but they don't really address the problem of is just, you know, that you're posing as well, right? Which is how do you avoid just keep on making it more complete?
Got
I
it.
think, in short, I think there's an answer to that, but no one's done it yet.
What do you think of the idea of discarding some information at each layer as you go forward?
Yeah, well, that's okay. So I wouldn't call it discarding. Okay, so let's go back to this. You know, I'm sorry, the Tai-Sing Lee and David Mumford diagram. So like in this hierarchical probabilistic model, the idea is that what you would send to higher layers just doesn't have all the detail. Okay, so there's things that stay behind in V1. Okay, so that doesn't mean they're discarded altogether, but maybe the details remain in V1, and it's more the abstract properties that are being carried forward to V2. So in that sense, these representations are complementary, right? If you want to know something about the 3D shape or whose face this is or something, then you would interrogate these higher level areas. But if you need to know detailed shape parameters, like if you're trying to take some tweezers and manipulate something like a thread, then you need to interrogate V1, right? Because that just, you know, it's not carried. That information is not discarded, it's kept around, but it stays in V1, right? And so that's the idea. So, and that you would sort of see, I think, with this kind of idea where this sort of condensation that I'm talking about, you expand, condense. The condensation or the contraction doesn't keep all the information, but that information, those details stay around in the lower level area.
But now the question becomes like, how do you know what to leave behind and what?
Well, yeah, okay.
Yeah, so thanks a lot, appreciate it.
Yeah, that's great. I mean, that's a great question, and that's why I'm, you know, I think, but that's kind of like the rough sketch of how we could talk more about it, the details of that. But that's kind of like the question you have to think about. That's right.
That's great. Thank you.
So, all right. So, in the interest of time, we'll stop the discussion for now. But you can, if you have questions, you can ask Professor Olsson again during the panel discussion. So, on to the next talk, Professor Jeff Christmer from UC Irvine. So, Jeff.
Great. Thank you, Hirak. And I'm the last speaker, I think, of the day, so it's been a wonderful day. I just want to say before I get started, all the talks and including the posters have been really interesting. So thank you, especially Hirak and Kashin for organizing this.
You can take 30 minutes. Sorry, you're starting late.
That's okay. We'll see. Just cut me off if I'm going too long, but I don't think I will. So I'm going to talk about motion processing and the primate dorsal visual stream. And it nicely, I think, complements Bruno's talk, the last talk. And really, this is work by Kashin Chen and Michael Baylor. And there's a paper coming out on the work I'll kind of towards the end of this will be talking about that'll be coming out very soon in Journal of Neuroscience. So let's get into it. Like Bruno was just talking about, sparse efficient coding. So the nervous system is under really tight energy constraints. So you have to represent efficient information efficiently. And I think this is particularly important for motion information, complex motion patterns, that you have to respond rapidly to those and appropriately. And I'll give you maybe a real world example why that's the case. Oh, I didn't want to start there. So if you're being chased by dinosaurs, you really have to process the motion so you can avoid things. And there's tons of motion, not only the dinosaurs themselves, but your own. And probably more day-to-day things are very important to respond rapidly and appropriately. And where does this happen? Well, the dorsal, quote unquote, wear stream in visual cortex is important for handling motion cues. And just kind of give you a brief overview, you have information coming in from the eye, it goes through the retina and the thalamus, and then to primary visual cortex, which was just talked about. And the areas that I'm going to talk about are area MT, which processes very local motion cues, retinal flow, optic flow. And then that projects strongly to an area called MST, which responds to much more complex motion patterns over a larger field size. And like I said, this has to be happening very quickly and very efficiently. And then those project to decision areas like the parietal cortex and other areas. So how does this happen? And I don't know if dinosaurs, I very much doubt, given what I know about dinosaur neuroanatomy, that it has a visual cortex, but I'm sure the dinosaurs had to have something, a homolog to this to actually process motion information quickly, given their behaviors in Jurassic Park. Okay, so two ideas that come out of this to reduce the amount of processing and energy are sparse coding and also dimensionality reduction. So if you had a completely sparse code, it'd be local. And you'd have these things like grandmother cells, but you wouldn't get much reuse out of this. It could only encode represent one thing. If you have a dense code, you're utilizing a lot of neural activity. So somewhere in between, you want a sweet spot that's sparse. And another concept is the difference between holistic, putting together a whole face, or parts-based, by reusing parts of the image or parts of a scene to actually reconstruct information. And so both of those ideas are energy efficient and reduce the dimensionality of the problem. And a few years ago, Michael Baylor did some very nice work looking at a machine learning technique called non-negative matrix factorization. And he took, instead of what was originally used for earlier was building up reconstructing faces, he took optic flow information and had a bank of filters that responded very much like area MT on the local flow fields. And then he injected that into non-negative matrix factorization. And lo and behold, you got, these are the weight vectors from the original flow image, MT image, to this matrix here that we're calling similar to, or has homologs to MST. And you see these weight vectors that look very much like MST receptive fields. And this is a nice way to do dimensionality reduction because you have a large feature space of all the different speeds and directions, and you have a large library of stimulus exemplars, and you can take this very large matrix and reduce it by a smaller number of basis vectors times the feature and the small number of basis vectors across the stimuli. And from that, and you see, and hopefully you see my mouse, a sparse number of neurons or a sparse number of units are active, and that's sufficient to reconstruct this image. So this kind of gave us an idea that if MST has these complex responses that fall out of this, enforcing this dimensionality reduction technique for machine learning, that maybe that's something that the brain is doing. But at this stage, it remained to be shown how you could do this in something like a neurobiological circuit with something that is more biologically plausible to do the equivalent of this non-negative sparse coding. And this is where Kashin Chen actually took this idea and made it more biologically plausible. So she implemented a spiking neural network model, and she evolved the spike time independent plasticity and homeostasis parameters. And I'll explain exactly what those are if you're not familiar in a moment. But let's just look at the kind of overall architecture. So you have, again, flow patterns, and you have a bunch of MT neurons that have different directional tuning and speed tuning, just like real MT neurons. And these are now spiking neurons. And they project with plasticity to an excitatory inhibitory area that we're calling MSTD. And from the weights here, we can reconstruct, or we can get a pattern that initially will not be like the original pattern, but what we can do is use an evolutionary algorithm to have a fitness function, and the fitness is how close the output here, the prediction, matches the original flow pattern, actually the original activity of MT to that flow pattern, okay? And this is a very large, can be a very large weight matrix. So rather than evolving the individual weights, which would take a long time and be a hard optimization problem, we optimized the learning rule. And there's a theoretical reason for that, but I'll get to that later on. So what we optimized, or she optimized, was the parameters for STDP, which if you know STDP, if you have the presynaptic activity before the postsynaptic activity, you get LTP. If you have the opposite order, then you have LTD, long-term depression, long-term potentiation, long-term depression. And what dictates how much potentiation or depression you get is the amplitude of this curve and the time constant of this curve. It starts to fall off if those two events are further away in time. And then there's, for the depression part, there's an amplitude for the minus part of the depression and a time constant for that. So for each projection, there were 4 parameters. And we had found that STDP on its own has stability issues. So we and others have added homeostasis kind of function, in our case, synaptic scaling, that pushes the weights, the overall weights of a projection to a target firing rate for the postsynaptic neuron. All right, and that's what I'm calling STDP-H. And to give you some details of what was done, we took a spiking neural network. We were evolving those parameters. So, and we had 50 separate instances or individuals. So we had to run this in parallel on a simulator called CarlSim. And we generated a whole bunch of flow fields and randomly held out some for validation and did sort of like a Monte Carlo as you go through different generations. And I won't go into the details of this. We can talk about it later if you want specifics, but there were only 19 parameters per individual that you had to search for the ideal values of, and it ran for 30 generations. And we used a form called evolutionary strategy that we can talk about a little later. And the bottom line is, with different sizes of MST pools of neurons, we got very good fits. So these are basically the correlation between the original MT pattern and the reconstructed MT pattern. And the B equals 64 means there were 64 neurons in the MST population. And you could see correlation values that are very high once you get beyond like a small network of 16. And then on the right, you see the learned or evolved STDP parameters. And if you know anything about STDP, this has a very biologically plausible shape as well, where you have a larger area under the depression and fairly tight time constant of the drop off, especially for the potentiation of how quickly it goes down. And then also if there was STDP on the inhibitory connections and the signs of the curve are flipped, which makes sense also biologically. So the bottom line is, under all these cases, it seemed to converge to similar solutions of these STDP parameters. And I'm not showing it, but also the homeostasis parameter firing rates. So we got good fits. This is just a representative example to show you what comes out of it. So you have the original pattern, you have the MT to MST connection weights, and you see these complex patterns that are spirals, that are translation, that are expansion, very similar to what you see in recordings in MST. And you see the activity to reconstruct which neurons would be active, and this is an example with 64 MST neurons. And that's a fairly sparse code actually to reconstruct the original input. And we did look at the sparseness. So we looked at, I just showed you an example of a population sparsity with the different size MST populations. And also there's lifetime sparsity, shows good lifetime sparsity as you get to more and more neurons, but not too many. That over all the stimuli that are presented to it, you get a subset of neurons, the neurons only firing for a subset of stimuli. So both population and lifetime sparsity come out of this, emerge out of this model. And after the model was trained and tested, then we gave it data sets from actual studies where they recorded from this brain area, MST. And so we used now new stimuli that were used in recording from monkey MST. For example, this work by Takahashi et al. And they found single neurons that responded to rotation and translation, having one neuron responding to multiple things of rotation and translation. And we also found neurons in our model that responded to rotation and translation. Looking at the total population, they found neurons that responded to the azimuth for lateral motion. I think the way to read this is more neurons responded to 180 or 0360, which would be the lateral motion. But if you look at the azimuth, I look at the elevation, you have more neurons responding to all elevations. And we saw a similar pattern in the spiking neural network model for this as well. So it nicely shows some of the responses and population responses and also individual neuron responses that you would see in real MST. Looking at another data set by Gu et al. In this case, the arrows mean the direction of selectivity of MST. So they found more neurons responding to when the monkey perceived moving forward, and these bands on the left and right are when the monkey perceived moving backwards. And if you looked at the direction selectivity of our model, you see the similar pattern. So it is picking up, given the inputs that they used in this study, the model is also picking up that kind of selectivity. So it's responding very much in that way like MST. And then the question is, well, what would you use these patterns for? Well, it seems like from MST, you'd also want to have a kind of behavioral parameter like encoding the heading or the focus of expansion. And so if you see this cartoon drawing of a monkey moving through space with its head turned and its eyes rotated, as you're moving through space, the optic flow will expand, and the focus of that expansion would be where the animal was heading. And so how well, given a pattern like this, can the model predict the focus of expansion? And so we used a linear estimator to take the weightings from MST to a couple of parameters to try and learn the XY coordinates of the focus of expansion, generate a bunch of optic flow fields, and did a whole bunch of 10-fold cross-validation. And comparing it to a previous model that did this with templates for the MST, like just picking out things that they thought MST would do and having templates, the spiking neural network model did much better as far as its focus of expansion error and for the X and Y coordinates. And this was not just with 60 size of MST is 64, but with across all the populations we looked at. So this also is an efficient way of maybe encoding and then decoding behavioral parameters like focus of expansion, or I can imagine some other important motion cues that you could use this representation to actually encode. All right, so let me wrap up with some thoughts about what this means. First, just what we did was we demonstrated a model of MST was able to account for a lot of these properties observed in monkeys. And we've done that now with non-negative matrix factorization and now with a spiking neural net that has a biologically plausible learning rule. And you find tuning to spiral motions, you find 3D translation and rotation, and you find encoding of perceptual variables such as heading. And I would imagine others if we do that. Now, the MSTD neurons learned a compressed and efficient and reduced representation of self-motion. And we were fairly agnostic to this. We just had it learn, evolve the parameters of spike timing dependent plasticity plus homeostatic scaling. And so we are operating under the hypothesis that maybe this is what STDPH is doing in the brain to actually get these efficient, reduced and sparse representations. And maybe other neurobiologically plausible plasticity could also be doing this. But STDP and homeostasis seems to have a very good way of actually kind of cutting out what's not needed, and the scaling also keeps the activity within range. And I think in general, this is a novel modeling approach that rather than hand tuning, manual tuning, or rather than searching over weights, you can evolve the learning parameters. In a complicated situation like this was kind of the only way we could actually come up with a solution. And so it's basically learning to learn or evolving to learn, which I think is an interesting concept as well. And lastly, going back to what this conference is about, or this workshop's about, what can computer vision learn from sparse reduced receptive fields? Well, real neurons spike. So why do they spike? And I've had arguments with people over the years, but I think the main reason neurons spike is to conserve energy, and that's not lost on a lot of hardware engineers. So they spike and they don't spike too often, and between spikes, they're not taking as much energy. But another important thing, especially for, I think, this problem of dealing with complex motion patterns, is they respond in a very timely manner. They respond very quickly. So sparse reduced spiking representations can conserve energy by reducing the number of action potentials, and I should have said also reducing the number of neurons needed to respond to stimuli. And this would be critical for many vision tasks. And just getting an idea, this is not lost on neuromorphic engineers, and this could be embedded because it's in this spiking domain and take advantage of neuromorphic hardware. And I'm showing not this model, but when we were in Telluride a number of years ago, we were working with the engineers from IBM True North at a very early stage for their chip, and we put their chip on one of our robots, and the convolutional neural net was just trained to learn to stay on the path and not go over the edge and down the mountain. And what's striking to me about this was the robot was using a small nickel metal hydride battery to run, and they hooked up the True North to that, which was running the convolutional neural net. And so this closed system was using very little power. We ran this up and down, and this is a very steep trail. It doesn't show in the video, but we ran this up and down this trail for about an hour on one battery without having to recharge and running everything. So I think, and Bruno was talking about this too, I think this is thinking about things in this way and having this pathway and looking at some of the tools to actually get these low power, small, what do we call it? Swaps, small weight and power systems that can be used in embedded applications, I think is very important. And it also may say something about what the brain is doing and why it's so energy efficient and why it evolved to have these energy efficient representations. So I will stop there, except to highlight the people that did the real work, which is Ka-Shing Chen, did all the spiking neural network work, and she worked with Michael and I. Michael did the original non-negative matrix factorization idea, and then she worked closely with Michael to get started, and then she really took off to do this work, and of course our funding agencies. And I will stop there and take any questions. Thank you for your attention.
Thank you, Jeff. That was a great talk. Is there any questions?
Yes, I have a question.
Okay.
Yeah, hello, Jeff. Yeah, the very last talk. I like you use the motion processing in MSTD and a spiking network as an example to study some general principle. So actually, my question is a little bit beyond what you have talked in your talk. So I noticed that you cite the Takahashi's result, and I also know like MSTD is not just a visual area, but it also a multisensory area. It could use the visual and the vestibular input to do the motion processing. And I know in his work, he found some neurons, which show opposite preference of the rotation. So I'm wondering whether you have some thoughts about this result, or your network model is able to explain that.
Yeah, I have thoughts, and our network model cannot explain that, but that's actually a very important point. MST is multimodal, so it is getting vestibular information. We didn't have vestibular information to put into this model, but that is extremely important for the problem too, because you imagine while you're moving through space, you're also getting vestibular information. And sometimes it was either Takahashi or it was another study. There's very few studies that can do this. Sometimes you can confound that, where your visual motion is not actually the same as your actually body motion, and you get interesting results. So that is something I would like to build in, especially if we did embed this model on a system that was moving through space, like a robot or a drone. I think that'd be a very important cue to take into consideration. Yeah.
Okay, thank you.
Can I expand on that, if you don't mind, Jeff?
Sure.
As I know, so in Greg DeAngelis and Dora Angelaki's lab, they're still studying that. And one idea is that these neurons with opposite preferences, visual versus vestibular, could help you with flow parsing to kind of disambiguate which flow is from your own motion and which flow is actually from an object moving independently of you.
Oh yeah. Thank you. That is a good point. So you're moving, but objects are moving, and so like if you want to disambiguate your own movement from some object that's moving, like that dinosaur, then that might be a way to actually make that calculation. Yeah.
Sure.
That idea was actually first proposed by Faison and Warren more than a decade ago. now people are finding the same concept in many different, with many different neural data flippers.
It's hard to do. The setup that Michael is talking about, as I recall, is you have a monkey strapped to a chair, seeing a flow field, and the chair is moving just back and forth. It's not even that natural. So, because if you're recording from these things, you can't have the like tons of movement around, so at least right now. So there's very few groups. That might be one of the only groups that is actually doing this kind of study and getting this information. Although I just saw a couple of days ago some amazing talks on fruit flies, Drosophila, and they are starting to get at some of these things because they can.
Is there any other question for Jeff? If not, then we'll move into the panel discussion session 2. So Cashin is at like a location with not very good internet connection. So she is actually the host for this session. So she sent me the questions as a host. So I will ask, but all the other participants like virtually or in person can also ask our speakers any question they might have either related to their talks from today, or could be related to their other papers or other previous research. Right, so. Yeah, so kind of open-ended question, since like we have very experienced researchers here, and then in CVPR we have so many young budding researchers want to pursue your career. So have you received skepticism or criticism when you introduced biologically plausible computer vision solutions to the CVML community? If yes, then what was your response to those comments? Anyone can take it.
Well, maybe just to clarify, what would the criticism be? Or to the person asking the question, if they can maybe chime in.
Well, it's a tough sell. I know what Hirak is talking about. There are some that some people sometimes, especially because I've gone more to the IEEE meetings, that aren't as interested in the biologically plausible ideas unless it shows something that they can use, which is a very much an engineering kind of point of view, which I totally understand. So one thing is to show an advantage. That gets tricky because the benchmarks for some of these systems are so optimized and done over and over again. It's hard to squeeze out something. So my advice and my approach is to try and find a new angle that someone hasn't looked at. That could be another direction that you could go, because you aren't going to compete like getting another percentage of a percentage point on ImageNet or something like that. But you might be able to show that, like what Bruno was showing, with the sparse coding, or some of the other talks showing things like motor reference, copy or attention or saliency kind of effects could actually improve in a different way. So that's sort of what my advice is to for that crowd to convince. Then there's the trade-off that you also want to convince the crowd that's the more biological crowd. So then you have to show that you can actually explain and predict something that they couldn't do from their experiments themselves.
Let's see, I think, yeah, I think I agree with all that. I know it is a hard sell in terms of trying to solve the problems that a lot of the computer vision community is solving right now. But I guess from my point of view, someone who thinks about biological vision a lot, that from my perception, like the computer vision community has defined vision in their own way as a set of problems that they can solve. with existing approaches, right? With existing hardware and things like that. Whereas the real problem of vision, like the thing that animals are solving with their brains, is just this monstrously complex problem that no one is even close to really understanding. So, I mean, that's the reason why I'm like doing these models, right? I'm trying to figure out the brain's problem. And so yeah, I mean, these models that we're kind of pushing on right now, they're maybe not, they look maybe somewhat wimpy for these, you know, for the standard benchmarks, but that's not the problem we're trying to solve. I would say, you know, for our own approaches, we're kind of trying to solve a different problem. So I think it depends on what you're trying to do, and you know what community you're speaking to, and things like that.
Yeah, and I guess like the example you gave in your presentation, the jumping, what is it? Jumping bee?
Jumping spider.
Yeah, versus the supercomputer. So like that little system is doing so many different things, tasks in machine learning terminology, versus where like in a CVPR paper, most of the time you'll find like a predefined set of tasks. So the skepticism could be the other way too. Like if you think about it in like a full system perspective rather than like specific task perspective. But I think most of the time, like Jeff said, the benchmarks are designed for 1 or 2 tasks, or at least one paper target 1 or 2 tasks. So yeah, so I think the objectives are different, maybe.
Yeah, and despite all the progress, and there's been tons of progress in AI and machine learning, it still is not comparable to organisms with a brain or a nervous system. So I still say that working in that realm, you've got a working model that is a path to actually getting what you call a truly intelligent system.
And maybe one more thing to add to that is I agree with what Jeff said, that it's hard to convince, and I actually don't think that the work we're going to do is going to add 1% on those benchmark performances, but I think it's the level at which we're contributing ideas. And I think one thing to always keep pointing out is that the most fundamental ideas that really spurred the deep learning revolution came from biological systems, right? And so I think you have to think about the level at which the ideas we're contributing from neuroscience can inform computer vision too.
Michael, how do you tackle those questions?
Well, I haven't gotten much pushback yet, but just give it some time and it will come. No, I was just thinking there are still, maybe one way to do it is there are still tasks at which the brain excels and that current artificial systems struggle with. And I think we've already heard about a couple of them, maybe in terms of robustness, failure modes, adversarial examples, but also generalizability, learning from small data sets, one-shot learning, zero-shot learning, that the brain is really good at. And I know it's being studied now, but maybe there's more potential to learn from the brain.
All right, so yeah, those are, I guess, all good points. Moving on, computer algorithms are usually seen as less biased, while humans have subjective feelings or opinions. Do you see any potential issues with making computer vision algorithms more human-like? If so, that probably means more biased, if like more subjective, let's say. If so, what do you think maybe some ways to alleviate the problems?
I mean, unfortunately, I think current computer vision systems just magnify the biases in their training sets, which are created by humans and therefore biased, right? So I think making these systems more certainly interpretable, but I think also human-like can actually help us interrogate those in a way that's
almost impossible with current systems.
Mhm.
You know,
Yeah.
it's a loaded question. I don't want to say biases, but I mean some of the cognitive biases, let's use that term. Some of the cognitive biases lead to behavior that's not rational, but there's very important reasons for survival that we have some of those biases. I'm not sure it's right to bake them in, but I mean that is why people act the way they do in many ways, and maybe that's important to get something that interacts with humans.
Yeah, so in a way, if we are designing an algorithm, some sort of human biasness is already there, right? Or generating a data set, learning from it.
Yeah, I guess another way to build off of what Jeff was saying is like another example is visual illusions, right? You probably don't, I mean, it seems kind of silly to put illusions into a computer vision system because those are often examples of how humans are fooled. But I think that those illusions are so interesting because they're illustrative of visual phenomena, right? So even though it kind of seems counterintuitive or like you're making performance worse, I think ultimately those insights will lead to more human-like and therefore better vision systems.
Yeah, and to follow up what Layla is saying, you just reminded me, I was on a project from an agency that was looking at some of the biases to understand why experts were doing what they were doing. So we all went to a lot of trouble, like using a scenario and building in biases to see if we could explain why they're making the decisions they were making.
Okay, next question. What's your usual thought process when developing a brain-inspired computer vision algorithm? Do biological constraints inform your algorithm design, or do you have an algorithm first, then try to find out whether it fits into, compatible with the biological constraints? What may be the benefits and or issues with either approach? So first, considering the constraints, then do you design the algorithm, or first just design the algorithm, then add constraints which are biologically plausible, and which way is the best? I think Bruno kind of talked about that.
Okay, I'll try to chime in on that one. I guess I would say the real problem is to define the problem that you're trying to solve in the first place. And so this is kind of what I was getting at earlier for, so like from the point of view of understanding the brain, then what you want to begin with, I think, is animal behavior. So how do animals use their visual systems? What did evolution design vision to do? And then start thinking about those problems. Those problems turn out to be very different than the ones that the computer vision community is solving, if you look at them really closely. And so, I mean, I think that's one big thing, is just to start out with defining what the problem is to begin with. Then you're trying to think about, well, how, come up with a theory of how that could be solved mathematically. And this is kind of was Mars' approach, sort of maybe not necessarily starting with a theory, but that's one way to sort of define the problem at that level and get it sort of a mathematical statement of the problem, and then drill down to some of the details, like how would I start implementing that? How can I express it in terms of variables that are interact with each other and so forth? And then, of course, then thinking about the neurobiological constraints. But I think it really begins with a definition of the problem. And yeah.
Yeah, anyone else? All of you design biologically plausible algorithms.
Well, I do a lot of robotics, so I do kind of exactly what Bruno just said. I say, what's the behavior I want to do, and I'm in this subfield called neurorobotics. So then, all right, so given that behavior, what brain areas are important for getting that behavior? And then I start thinking about the algorithms, but it starts with what's the problem I want to solve. Another way I've looked at it is, I'm really interested in this brain area. So what does it seem like this brain area does? What behavior and problem does this brain area seem to be solving? And then I start to build up, a behavioral experiment or going that direction, and looking as much as I can, like what are the inputs and outputs and connections and interactions of that part of the part of the brain. I'd like to hear what Michael thinks, though, because he's working in a whole other area of prosthetics. So what drives the way you actually kind of approach your problems?
Putting me on the spot. Yeah, it depends. I don't have a one set answer. Often, yeah, let me think of a good example. Well, okay, what I showed in my talk was really started with the mystery of how do you go from a stimulus to perception, and that covers the first step of problem, defining the problem, and then you would zero in on the brain areas it's involved in. It's basically what Jeff said, and you try to understand what are the computations that might be going on there that could give you an insight in how to go from A to B. But to contrast that, there are other issues that are more engineering driven. For example, if we want to do scene simplification in real time, we need a fast algorithm. We also need robust algorithm, and we need an energy efficient algorithm so we can put it on an edge device. And so then the problem is really more given like, how do you do object segmentation on a stream of spikes? Yeah, so then that's more, there it is more, I guess, about performance of any model that could be brain inspired, because maybe we need to look to the brain in order to figure out how to do it. I don't know if this helps, yeah.
What about, I mean, you're dealing with people that are dealing with clinical patients. I mean, there must be some design decisions or order or things that you have to take into consideration when you're building your prosthetic devices.
Yeah, I'm a proponent of that. I feel like it hasn't been done enough. So there are like two fields: there are like clinicians, and then there are engineers who actually built the system, and they don't always talk to each other as much as I would like. And so I think going forward, it's important to involve the end user at every stage of the design, so-called patient-centered design, to figure out is the problem you're solving, is that even a problem for the end user, or maybe the form factor you propose is not adequate. And so it's more of an iterative design process, I would think, a lot of back and forth rather than I have these set steps and I just apply these every time, and that leads to success. Because sometimes, for example, in the with the NMF, that was more of a happy accident. I was messing around with dimensionality reduction techniques, and then I realized, hey, if you do NMF, you actually get something out that looks very much like MST. And yeah, that definitely happens.
So Bruno, when you were developing the sparse coding first idea, so was it from like power efficiency, less activation point of view, like the motivation, or was it from like a biologically, like designing how the brain works?
A little bit of both. I mean, actually, it was David Field who really pioneered a lot of those ideas. And, what he was trying to think about is, well, he has a very nice paper in 1994 in neural computation called "What is the Goal of Sensory Processing?" In fact, that's the paper that kind of made me want to go and postdoc with him. But trying to understand what are, can we state some kind of intermediate goal of these neural representations in the brain? So of course, the overall goal of the system is survival, how to survive in the world and have useful behavior and stuff. But how do we break that down into something that we can build? And so he was trying to think about these design principles, like, principles of neural representation. And then there was this recent work of Joe Attic and Norman Redlich on trying to understand what the retina is doing in terms of whitening. And so, there was all these ideas building at the time that maybe we can understand neural coding in terms of natural scene statistics, in terms of the statistics of the signal coming into the system and how to efficiently process those. So that's both a very attractive engineering idea. That's what any engineer would do who is building a compression system and so forth would do. But there seemed to be evidence gathering, at that time that this is something that biological systems are doing as well. I think that was kind of the hook, maybe the sort of the entry point that sort of drew us into that. And I think, maybe, that's to me, like maybe one of the most interesting aspects of this work is that when we find problems that we think both brains are solving and engineers either have solved or would like to solve, right? When those align, and then you find that biology and engineers are doing very similar things. For example, in the design of the eye itself, right? The optics of the eye, if you just look at how the optical system is built, the size of the eye and all these different animals, is just like an engineer would build it using optical engineering principles. And so the question is, is it going to be that way for the brain, for the nervous system? And I think we have a lot of evidence coming out now that it's very much looking like that. In these early stages where we can kind of have a chance of understanding what the goal is. And so I'm hopeful there's going to be more of that, right? And so we're going to learn things from studying the brain that are going to be new technology tools. They're going to be new kinds of computer science principles, new kinds of mathematics coming out of this. And just one final comment I'll make is that, the original McCulloch and Pitts paper, I was just, diving back into that from 1943. It's hard to appreciate at the time, but in their paper, they have these pictures of logic circuits, so how you could compute the OR function, the AND function using simple neuron models. Those drawings in their paper, those were actually the first logic gates ever. And von Neumann himself was very much inspired by McCulloch and Pitts's work. And that work was sort of pivotal for him in designing the first computing architecture. And so what's kind of neat about that whole story is that McCulloch and Pitts were thinking about how the brain works, right? It's just sort of like that act of thinking about what is the brain doing. That is an act of discovery in itself, right? It's sort of what I'm not active, but it's sort of a creative. It's a sort of it's something that sort of spurs the creative process, right? And they were able to develop something that was really a pivotal discovery. And so I'm hopeful we're going to have more of those kinds of things and this kind of synthesis between what biology is doing and what technology is doing. So counter to the question at the outset, which has always seemed kind of odd to me, like to receive criticism about, gee, why are you doing biologically inspired approaches? It's like, it's the exact opposite. It's like, oh my God, what else would you be doing? I mean, this is like the best thing you could possibly be doing, right? So I know, yeah, anyways, that's my view on it.
Yeah, engineering, like the silicon brain. One maybe last question, almost out of time, to Professor Isik. So do you think, like with the current approach, we will be able, like following the current approach, we'll be able to teach neural networks about context or common sense, like the example you showed?
No, but I think that there's a lot you can add to current neural networks, like a lot of, like the graph structure, or other sorts of inductive biases, or the things other people have talked about, like iterations on learning rules and things like that, that will get us a lot of the way, but not to full human cognitive reasoning. But I think they'll get us a lot of the way in terms of seeing.
All right, so any other questions? So we have, not many in-person audience, but we're really thankful that you're here. Or any other question from the online attendees? If not, then I guess we can go to the closing ceremony, give the awards. I mean, no one is here, so we'll just name them. Jeff, can you do that?
Yeah, I can do it if you want. So the judges went through the posters and picked two. There was agreement across all the judges for two posters. So we're going to give out two awards. The first one goes to poster ID 4, Neuromorphic Foveation Applied to Semantic Segmentation by Amélie Gruel and colleagues. And the second award goes to poster ID number 5, Learning Hierarchical Temporal Representations via Dynamic Predictive Coding by Preston Zhang and colleagues. And yes, congratulations. Actually, I went to as many posters as I could, and I looked at the lightning talks ahead of time and today, and they were all really good. So, so thank you. Thank you, everyone, for your contributions. And you awardees, you'll be hearing from me with your award, which would be I guess the easiest thing to do is electronic gift card is what we decided. And do you want to say anything else, Farak, or you want me to close?
Maybe one last reminder is that there is actually a follow-up. There will be a follow-up special issue in the Journal of Biological Cybernetics. So it will be on the same theme as this workshop. So submission is open and will end on September 30. I hope all of you attending here can contribute. And yeah, with articles or any capacity. So I think that's all. And any other.
Let me say one last thing is the whole idea of this workshop, I think came originally from Hirak, and he's worked really hard on this. And then Kashin got really excited about it, and the two of them not only got the amazing cast of invitee speakers, but they also found the right venue to have this, and then all the work that went into the planning and executing of this. I think you guys did a great job, and so thank you for that, first of all. And
thank you for hanging in. I mean,
yeah.
It was rejected in a couple of places. So
yeah, it's been a long day and lots of time zones were involved. So thank you, everyone. And yeah, do consider submitting something to this special issue. This is a really good journal, and I've participated in special issues of these two in this journal too, and that they really they put together a nice thematic journal issue for some of these things. So do consider to contributing to the special issue and following up with some of your great work that you did presented today. That goes for everyone, or something you want to do that's along the lines of what can computer vision learn from visual neuroscience.
All right, so I guess that's it from my side. Jeff, do you have anything else to add?
No, I hope to see you all again, either virtually or in person.
Yeah,
thank you, everyone.
Kashin is unfortunately in some bad network area, so yeah. And then we also have Xiumin Li from Chongqing University in China, who's also a co-host. So thank you all of you, and thank you for joining, and have a good night.